Okay, good morning, everyone. Thanks for being here. And first of all, just to recognize our leader, Anna Andriazza. I think one thing that I've learned from Anna, that in addition to her excellent science and leadership, she always keeps her eye on the patient and the families. And we have Kate Murray here, who's the CEO of Mito Canada, who really is that fantastic linkage we have to the patients and their families. So thanks, Kate, and thanks for that really great kind of perspective that all of us need to have. So I think I know that Soumya introduced me as a surgeon, and yes, I am. So I just have to tell you that I take a little bit of a surgical perspective to a lot of things that I do. One of the things, the focus in our research laboratory is really around liberally using animal models to study disease and the biology of disease, and then going back and using that to innovate new therapies and also to understand why the therapies may or may not be working. And I think what I'll do is I'll mix in a number of those things together when I talk about our work on mitochondria and liver ischemia reperfusion injury. So earlier on in my career, I was a trauma surgeon. I couldn't stay up overnight anymore, so I had to give up trauma surgery. But one thing I didn't forget was actually the impact of hemorrhagic shock. When a patient came in profound shock from a major trauma to the hospital, it was pretty clear that related to the combination of ischemia from the low blood and the subsequent resuscitation that we did, that patients developed something called ischemia reperfusion injury. And that's really been the core of the work that we've done over the years. I think it's very well known that ischemia reperfusion leads to oxidative stress in the cell. It's also well known that that oxidative stress in and of itself is deleterious, but in addition can lead to the induction of a number of pro-inflammatory cytokines, as those listed here. And then together, those bring neutrophils into the organ and actually see what we see in patients who are in the intensive care unit, which is a systemic inflammatory response, and in addition, multiple organ failure. And I think that those are the areas that we work on, predominantly ischemia reperfusion injury. Now, this is a schematic of a mitochondrion. And every time I'm with my graduate students, I say, don't ever use this schematic. It's so confusing. Nobody will ever understand it. But Dr. Lin used one, so I feel like I'm empowered to use it. But it is actually a little bit helpful sometimes. So on the left, you'll see the schematic of the mitochondrion, the ischemia, reperfusion having an effect on the mitochondrion, in essence, initially depolarizing. And without going into the details, I mean, I think that four major things that occur in response to ischemia reperfusion is ATP depletion, release of mitochondrial DNA, which can be in itself deleterious in terms of having a pro-inflammatory effect, apoptosis, which we talked about already, and the release of reactive oxygen and reactive nitrogen species that themselves are deleterious, but also induce a profound inflammatory effect. And I think that that's the effect of ischemia reperfusion, and that's why I think it's pretty clear that if you can do something to modulate mitochondrial function or augment it, that you might be able to have an important positive effect. So our overarching hypothesis through the work that we do is that mitochondrial dysfunction is a hallmark of liver ischemia reperfusion injury, and that if you can restore that somehow, that you might have a therapeutic benefit from it. And in our group, I'll just talk about three things that we are doing. One is augmenting mitochondrial function through pharmacological means, and those of you who were here yesterday saw my student Lillian Huang talk about AP39, which augments the Krebs cycle. Previous work that we did in patients and in animal models where we gave us ischemic preconditioning, we showed that induced increased mitophagy, and the clearance of the dysfunctional mitochondria was responsible for the improved effect that we saw with the intervention. And of course, we're here today to talk about mitochondrial transplantation. And what I'll do is I'll hopefully be able to show you some of the work that we do. So I have to recognize my former PhD student, Avinash Mukala, who is now in San Diego doing his postdoc for much of this work, but really it's a team effort. And we actually use a number of models, and I'm going to take you through some of those models, including some of the mechanistic things we do, and I think end up a little bit near where I think Marcello talked about at the beginning, was really the ability to transition our transplants that are suboptimal into humans so that we can have a beneficial effect of using suboptimal organs. So here's the model he used. It's a pretty standard ischemia reperfusion model of the liver, where you basically make the left lobe ischemic. I think that after you have it ischemic for approximately 30 to 60 minutes, you reperfuse the animal. And that's where we actually gave, during the reperfusion phase, the mitochondria. So in this model, actually, we inject the mitochondria directly into the spleen. For those of you who know kind of animal anatomy well, and human anatomy, injection into the spleen will get you a direct conduit into the portal vein and into the liver. So very effective way of delivering mitochondria into the liver. And I'll show you some of those pictures later. And we did a series of evaluations. I think you can do all the evaluations you want, but what you really want to see is that if you give animals IR, you markedly increase the release of hepatocellular enzymes, which are indicative of necrosis. You markedly increase it by IR, and you can see in the block that's circled in red, you get a marked improvement in the AST release, and ALT release goes back to near normal again. And the controls are on the right side, but I think relevant to what I think Marcelo talked about, we showed that non-viable mitochondria did not have an effect here, so it had to do something clearly with the viability of the mitochondria. We also showed that the beads and various other vehicles were not effective here. So viable mitochondria injected into the portal vein via the spleen caused a marked reduction in the hepatocellular enzyme release into the blood, and that was actually mirrored in addition to that with histology that showed an improvement. And one of the other things that we did was we looked at the inflammatory response, and I think there are two things to take away from this. Whether you're measuring basically IL-6, tumor necrosis factor, both highly pro-inflammatory, mitochondrial infusion actually markedly reduced the release of those pro-inflammatory cytokines. But very importantly, and I think central to some of the hypotheses that we're addressing now, if you looked at IL-10, which is an anti-inflammatory cytokine, the infusion of mitochondria markedly augmented the IL-10 release, suggesting that we go from a pro-inflammatory environment induced by IR to one that's profoundly anti-inflammatory. And what was interesting as well is this effect was not only true in the liver, but actually at distant organs as well. So infusing mitochondria into liver had a profound effect throughout the animal. So I want to talk about those things I talked about before, which is reactive oxygen species, ATP. Here's an example of measuring liver and systemic oxidative stress. Again, by restoring mitochondria into the liver, you markedly reduce that oxidative stress, and not only in the liver itself, but also systemically. If you look at ATP content, you can see in the blue bars that giving mitochondria actually preserve the ATP levels, which is another potentially deleterious effect of ischemia reperfusion. And then finally, I think it's really important to study exactly where those mitochondria go. And I'll show you a series of short videos to show that. So on the left, you'll see an animal that actually only receives mitochondria. So we tagged the mitochondria, and we could follow them by in vitro microscopy, working with Paul Koobs at the University of Calgary. On the left side, you can see that's the flush of mitochondria, first mitochondria going through. You don't really see very many sticking. So if you take in contrast to that, the flush of mitochondria, you can see there's a profound sticking of the mitochondria within the liver. And it's actually throughout, and it's mostly associated with the blue cells, which are actually tagged, which are Kupffer cells here. And you can see in that bar on the right that there's about 25% of the Kupffer cells that we monitored actually now had mitochondria associated with them. So just to give you a little bit more of a close-up. So that's a close-up of a mitochondria, and here are the mitochondria actually adhering to the Kupffer cell in advance of them being taken up and having their effect. A couple of things came out of this kind of study. First, we understood that the mitochondria were almost entirely going to the Kupffer cell, and Kupffer cell was probably mediating the effect. But the other thing is that clump that you're seeing is not one mitochondrion. So what we believe is that actually mitochondrial clumps that we work so hard to get rid of when we're preparing the mitochondria actually may be the type of way to deliver that. And I've talked to Frank Gu and tell him that we have to have clumps from now on. So it turns out that the Kupffer cells are very important in the effect that we want to see. So you can see that if we do genetically take out the Kupffer cells, you can see that we no longer see the protection. And I think that's best shown in the upper right, where in Kupffer cell depleted animals, the protective effect of mitochondria on the AST release is totally gone. And if you look at the lower part, all the protective effects seen with mitochondria in the inflammatory cytokines is also gone. So Kupffer cells are critically important here. And one of the things we wanted to monitor was how, we heard a little bit how mitochondria get into cells and how they integrate. So this is a study that actually looks at the pH of labeled mitochondria once they get into cells. And if you compare left to right, you can see that those mitochondria that get into the cell really are markedly acidified, which suggests that actually they're somehow going into the endosomal pathway, endosomal lysosomal pathway, and potentially being degraded there, rather than necessarily integrating directly into the mitochondrial network, although we don't have evidence for that. So I just want to also add in work we did with Sonia McParland at UHN. I think a talk wouldn't be complete unless you showed RNA-seq. So here's my picture of RNA-seq. And I just want to point out that we looked at all different types of cells, but we really have an interest in Kupffer cells that I showed you already. But there are also a lot of other macrophages that are in the liver that are not Kupffer cells that actually come in response to the injury. And so we are interested in looking at both those types of cells and what they do. Our hypothesis is that Kupffer cells were profoundly anti-inflammatory, but maybe they had an effect on these other Kupffer cell populations, monocyte macrophage populations in the liver. And here's just one example of that. So we developed a score. One score was our inflammatory score. So our anti-inflammatory scores use markers of M2 to M1. And so you can see that those are genes in the bottom that are basically have M2 or M1 as sort of their predominant type of gene expression. And you can see on the left, the M2 to M1 in the Kupffer cells showed a significant increase in the anti-inflammatory profile by the Kupffer cells, but none from the monocytes or macrophages that are in the parenchyma. By contrast, if you look on the right, and you look at kind of genes associated with efferocytosis, which are genes that actually lead to clearance of dead tissue and other apoptotic cells, you can see that not in the Kupffer cells, but in the monocyte macrophage populations that's in the parenchyma, there's actually an increase in efferocytosis genes in those cells. So we think that probably the Kupffer cells are modulating the effect, and they're having an effect by their release of anti-inflammatory cytokines on on monocyte macrophage populations that are in the parenchyma. So this is early stages, but we have a lot of work to do there. I want to talk about how we're translating it now to larger animal models. So obviously, we want to go small animal models, porcine, and then maybe humans. We did studies with Marcus Selzner and his group at UHN on looking at a porcine model, and you can see here, there are a lot of things that we measured, but the key part is we did 2 hours of hemi-liver ischemia, followed by a reperfusion period. We again put mitochondria in at the beginning of the reperfusion period. Because the portal vein is big enough in a pig compared to a mouse, we actually directed directly injected into the pig. And I'll just show you a couple of summary slides. So this is a summary slide of what we saw when we gave mitochondria in this liver IR model. You can see in the red there's a marked increase in the plasma AST, and that's abrogated by the use of mitochondria. Similarly, if we did histology and we looked for a reduction in necrosis, that mitochondria actually significantly reduced the amount of necrosis in there as well. There are a lot of kind of indicators of liver function in general. One of them is the ability to support coagulation because it produces coagulation parameters. Again, on the left, you see that we actually preserved the coagulation parameters by giving mitochondria, and on the right, the anti-inflammatory effect was seen. And then finally, we were working with Anno Heise, who you'll hear all about his work later on today. I was telling him I was going to talk all about the physics of photoacoustic imaging. I know nothing about the physics of photoacoustic imaging, so you can relax, Anno. That's okay. But so you can see on the right of the screen, you can see a graphic that shows the change in oxygen saturation that he measures. And in the blue and purple, you can see there's a marked reduction when you clamp the liver. That was what we expected to happen. When we infused saline during the reperfusion period, we actually still had a reduction in oxygen saturation. So the liver clearly wasn't doing very well. By contrast, on the left side, you can see again we saw a marked reduction in the oxygen saturation, but by adding mitochondria in the perfusate, we actually had a move back up towards the normal levels again. So it's not only working on an anti-inflammatory effect, we think there might be an effect on endothelial cell function or various other tissues that may be modulating the flow there. So finally, I want to close with something that's really, I think, a follow-up with what Marcelo was talking about, was this model of ex vivo liver perfusion. So I think, Marcelo made a great case for the fact that using ex vivo perfusion as a conduit to transplantation would be an excellent way for preserving organs longer in order to allow you to transplant them safely. It also allows you to have strategies that might improve the quality of the organ before you put it back into the patient. So we've done with Dr. Selzner's lab basically a series of studies. We actually took this picture yesterday, so I can show you this is actually the porcine liver sitting in a bowl. That little blue area that you see at the bottom is actually the tube going into the arterial side of the liver, and then we drain out the effluent on the other side. So we are, in our series of studies, are trying to kind of do studies that look at optimizing the use of a DCD organ for transplantation. Here's our protocol, and it's interesting, we're still using 4 degrees centigrade of storage, so now that I saw all those kind of references, I'm going to maybe go to 10, but basically in this model we're giving DCD, which is basically warm ischemia for an hour. We're storing the liver for 5 hours, but under this perfusion situation, and then we're taking a variety of parameters from that after we start infusing the mitochondria again. So this is, we're at early stages in this, but we're, hopeful that we're going to see a similar effect in the ex vivo liver perfusion as we've seen in the liver IR in the pig as well. Obviously different models, but probably needs a little bit of optimization, but that's the model. I think early on, I think it's pretty clear to say that, using EVLP, that you might lessen it. We've shown that you've lessened injury a little bit. We've improved blood flow there, and we know that fresh mitochondria are superior to those stored at 4 degrees for 5 hours. So that's our initial study. We're going to kind of do a fair bit of work. That's what we want to do with our Mito2I grant to understand better what that is. And I want to close with just a comment on our ongoing studies. So we're spending time now looking at optimizing the EVLP model in pigs. in preparation for transplantation back into pigs, but really with the expectation ultimately we're going to be able to do this in humans. I think we're really focused at the same time on studying the mechanisms of the beneficial effect of mitochondrial transplant and investigating different mitochondrial preparations, both in vivo and in vitro, which is something that I think our team is working on. You're going to hear a little bit about, I think, organoids later on today. I think Hagar is going to talk a little bit about, delivering mitochondria and maybe in an organoid system. The one thing that we've started to do now is something called precision cut liver slices. So basically you take a liver, you take a piece of the liver, and then you process it. And you process it through cutting it in slices that are thick slices. You culture them in vitro, and you can keep these slices alive for about 14 to 21 days. So that suggests the possibility that you might be able to study different types of preparations of the mitochondria in these models, and actually what is a really good representation of the 3D structure of the liver. So we like that. And on the right side, that's actually taken out of a paper we just submitted. It's basically using, looking at tagging of mitochondria with a dual GFP M-cherry tag, and you can see that, when you add in after 14 days these vital dyes, basically that you have basically mitochondrial structure there as well. So this is a model that we may be able to use, combine it with maybe perhaps a perfusion model so that we can control the oxygen saturation, the flow, various other components we want in there in addition to the mitochondria, and measure that. The other thing that is really attractive of this model is that if you look on the left, as a surgeon, we look at liver all the time. And those preparations you see on the right are actually human liver. So we can take liver slices out, and you don't need very much, probably 2 or 3 cubic centimeters, and then basically take those and get what you see on the right. So thanks for the opportunity to present. Happy to take questions during the next phase, and I think the next speaker is set to come up. Thank you.